Hi, my name is Yasmin and today I'll be talking about plant electrophysiology. So here's my outline for my presentation. I'll be walking you through the definition, what is signaling, what are the sources of it, what is ionic flux, what kind of information this can provide us, the methods for collecting such signals, what the data looks like, and then I'll be taking questions. So first off, what is electrophysiology? Electrophysiology is any electrical chemical phenomena happening in biological cells. This applies to any living biological cell, not just plants. And if we were to zoom in one layer deeper, what is electrochemical? It is the ionic balance generated and propagated for signaling purposes. What I like to focus on in this is signaling purposes, so it means any ionic shift, plus or minus. We're thinking about gradients, which we'll look in further. We'll create an electrical signal, and what we're focusing on in this presentation, or the core of it, will be electrical signals, which is the time-varying voltage. We can think about that as circuits. So what are the electrical signal sources within a plant? A plant receives a lot of nutrients, but also has and its cells all types of different ions that are flowing. So most frequent ones that will be creating the signal of interest will be protons such as hydrogen, potassium, chloride, sodium, and calcium. And less frequently, the ones that are the source would be transition metals such as iron, aluminum, magnesium, zinc, manganese, and mercury. Other sources can be nutrients because we know that they're being pumped through the roots, amino acids or organic compounds, things that we can find in the soil or also outside as gas form. So just to give a little bit more of an idea of this, when we're talking about what is traveling, and we mentioned the most frequent ones, well, it means it can also be intra or extracellular. So here's a picture of how they would cross through the cell membrane, and you will have plus and minus, and in this case we are seeing potassium that is binding to something else, and then you have sodium and chloride that are also binding. And this means that when they're going through the cell membrane, we will have gradients either of sodium chloride or potassium in this case. So these ionic fluxes are mostly focused on the three ones that we just looked at. And why? Because they have the highest mobility within a plant. They also are at highest concentration, so it's easier to map out focusing on these ions. So what would cause them to shift and go through the cell wall? We are thinking about osmosis, so any salt stress, a change in pH, a change in temperature, or any other external stimuli can actually affect the ionic flux or have a kind of snowball effect on reactions happening within the plant. So far we've been, just to kind of summarize where we're at, we are interested in ionic flux, electrical signals, which are action potentials that can be long or short, and we're talking about a singular plant. So I mentioned nutrients, so we're going to be looking at the root zone. It can also happen in the phloem or the leaves, and inter or extracellular. But there's one thing I'd like to make a little parentheses on, is that also we mentioned amino acids and things in the soil or any medium they're growing in. So the electrical signals can also occur from plant to plant, but when reading this, we have one signal. So it is a resultant of these stimuli. So what do the electrical signals tell us? Maybe it can tell us a little bit more about its environment, and that's what I tried to show in the last slide, is that it's any external stimuli can give that, and that external stimuli can also be caused by its same species. It can also be local stimulation. That's another example. So think about the Venus flytrap that is closing in when it senses movement near it. It can also be the resultant of reactions happening within, so any ionic flux shifts that are not also caused externally, but just regulation purposes within the plant. So we can think about respiration, And could this provide any insight on plant behavior? These are questions that people are asking when tracking this type of data. So the methods to collect data for electrical signals and better understand electrophysiology of plants can be invasive and non-invasive. And what I mean is sometimes you actually need to puncture the plant to understand its electrical signal. And these things are microelectrodes that you put into the stem of a plant or any part of it. So picture one, we can see that, or a patch clamp, which is another form of a microelectrode for collecting the electrical signal in the plasma cell. And then you have non-invasive, which is a surface contact electrode or metal electrodes. You can put them in the soils if you're monitoring around the roots or on the surface of the plant, and it would resemble what you would have for an electrocardiogram. The difference between intra and extra, just to make it extra clear, is intra will look at ion channels, and extracellular we're looking for signals from cell to cell. So we know that the signals will look differently. Once you've collected this data, you will have a signal that varies across the time domain. But when you have an electrical signal, it's important to know that it can be continuous or discrete depending on how you collect your data. So if it's analog or discrete with your sensors, and you can also analyze within the frequency domain. So just simply looking at what are the most recurring voltage that you will see, and what are the relationships to the other ones. What is this comparable on the human scale? I mentioned quickly the electrocardiogram or the electroencephalogram, mostly known as EEG, so what you put on your head. And why am I mentioning this is that a lot of noise will occur if we're trying to understand the environment, and there are reduction methods that can be used and transferred to analyzing plant data, such as wavelet transforms. So here are my references, and I'll be taking questions if anyone would want to take a question. Thank you.